Module 4, Objective 12, identify the structural and functional differences between skeletal muscle fibers and cardiac muscle cells. Cardiac muscle cells are only found in the heart. They're also known as cardiocytes. They're highly excitable. Their membranes are excitable to conduct the electrical impulses within the cell, and they're striated, similar to that of skeletal muscle cells. Unlike skeletal muscle cells, cardiac muscle cells are small. They're typically branched with a single nucleus. They have short, wide T-tubules, and they're without triads. The sarcoplasmic reticulum of the cardiocytes lack terminal cisternae, and they're almost completely dependent on aerobic metabolism. So they require a fair amount of oxygen for the mitochondria to generate aerobically. They contain lots of myoglobin, and remember, myoglobin is a protein that stores oxygen. And they connect to each other via a structure called an intercalated disc. These intercalated discs are specialized connections. They join the sarcolemmas of adjacent cardiocytes by gap junctions and desmosomes. The functions of the intercalated discs include stabilizing the position of adjacent cells. They maintain a three-dimensional structure of the tissue itself, and they allow ions to move freely from one cell to another. So cardiac muscle cells beat in a rhythmic pattern. Here we see the typical cardiocyte. Please note the intercalated discs located within this tissue in between the cells. Here's another view. Notice the wide T-tubules, and the sarcoplasmic reticulum is fairly different as well, namely in that they lack those terminal cisternae, those pouches that we see closely associated with the T-tubules. There's four functions of cardiac tissue. One, they exhibit an automaticity. They contract without neural stimulation. And their contraction and their rhythms are controlled by pacemaker cells. These are cells within the heart that actually establish the electrical activity of the heart. They have variable contraction tension that is controlled by the nervous system. They have an extended contraction time that is almost 10 times as long as that of skeletal muscle. And they're able to prevent wave summation and tectonic contractions by cell membranes. In Module 4, Objective 13, we look to identify the structural and functional differences between skeletal muscle fibers and smooth muscle cells, as well as discuss the roles of smooth muscle tissue and systems throughout the body. Smooth muscle tissue are found throughout the body. If you consider the integumentary system, the erector pili muscles are smooth muscles. And remember, these muscles, when contracted, actually erect hairs, both for thermoregulatory mechanisms. They can, by standing the hair straight up, they can actually provide a means of retaining heat. But they're also tied with our emotional systems as well. So if we were to become scared or upset, our hairs, our goosebumps can form. So those erector pili muscles can contract and goosebumps can form. We find smooth muscle tissue in the cardiovascular and respiratory systems. They help regulate blood pressure by changing the diameter of the blood vessel, as well as airflow. Digestive and urinary systems, they form sphincters, as well as move materials along and ultimately out of the body. If you consider the digestive system as a tube from mouth to anus, that tube is lined with smooth muscle, which help propel things through the digestive system. And lastly, within the reproductive systems, they help with the transport of gametes as well as expelling the fetus. Characteristics of smooth muscle: they're very long, they're slender, they're spindle shape, often described as cigar shaped. They have a central nucleus, a single central nucleus rather. They lack T-tubules, myofibrils, and sarcomeres, and they're non-striated, so they don't have the striated appearance that we see in both the skeletal muscle fiber as well as the cardiocytes. They have scattered thick filaments with many myosin heads, and the thin filaments are attached to structures known as dense bodies. Dense bodies connect cells to adjacent cells, and they help with transmitting contractions. Smooth muscle lack tendons, and they lack the aponeuroses that we see with the skeletal muscle fiber. Here we see the characteristic smooth muscle cell. You can see the relaxed state up here at the top, followed by the contracted state down here at the bottom. Functional characteristics of smooth muscle include 4 ways that smooth muscles differ from other muscles. Excitation, contraction, coupling mechanisms differ from that of skeletal muscle, as well as the length-tension relationships. Without the sarcomeres, the tension and resting length are not directly related, so smooth muscle cells can function over a wide range of lengths. Multi-unit smooth muscle cells are innervated by motor units, and each cell may be connected to more than one. Visceral smooth muscle cells are not connected to motor units. They're arranged in sheets or layers that exhibit this rhythmic cycle of activity controlled by pace setter cells. Lastly, smooth muscle tone. There's normal background levels of activity within smooth muscle which provide the muscle tone that can either be decreased by neural, hormonal, or chemical factors